When we say metabolism, the sum total of all chemical reactions and physical workings occurring in a cell. We have two types of metabolism, the anabolism and the catabolism. When we say anabolism is a biosynthesis, a building complex molecule from simple ones, or requires energy or ATP. Catabolism is the degradation, breaking down complex molecules into simple ones, or generates ATP or energy. So we have heard structures or the process of the synthesis of cell structures. That one, from energy source, carbohydrates, lipids, to catabolism, end product. Then the center is the ATP or NADH+. From single building blocks, sugar, amino acid, to anabolism requires energy. Proceed to macromolecules, carbohydrates, protein, lipids, then go to source of energy. We have here the enzyme structure. Simple enzymes consist of protein alone. Conjugated enzyme or holoenzymes contain protein and non-protein molecules. A apoenzyme is protein portion. The cofactor is non-protein portion. So metallic cofactors is iron, copper, magnesium. The coenzymes, organic molecules, is the vitamins. So we have here the pictures of that. So you can see that coenzymes, the metallic cofactors over there, and the enzymes, the coenzymes, metallic cofactors. So we have also the enzyme transstrate interactions. Exoenzymes transported extracellularly, where they break down large food molecules or harmful chemicals. Cellulase, amylase, penicillinase. Yun, that's it. The endoenzymes retained intracellular and the function there. So we have the transstrate and substrate and the product. Constitutive enzymes always present and always produce in an equal amounts or at equal rates, regardless of amount of substrate. Enzymes involved in glucose metabolism. Induced enzymes not constantly present, produced only when substrate is present, prevent cell from wasting resources. Synthesis or condensation reaction. It is an anabolic reaction to form covalent bonds between smaller substrate molecules. Require ATP, release one molecule of water for each bond. Hydrolysis reaction. It is catabolic reaction that break down substrates into small molecules. Requires the input of water. So we have here the image, the glycosidic bond and the peptide bond. Transfer reactions by enzymes. Oxidation reduction reactions to transfer the electrons. The amino acid aminotransferases convert one type of amino acid to another by transferring an amino acid group. Phosphotransferase transfer phosphate group involved in energy transfer. The fourth, the methyl transferases move methyl group from one molecule to another. The carboxylases remove carbon dioxide from organic acid. So we have the metabolic pathway, the multi-enzyme system. See. So we have heard the control of enzymes activity. First, we have competitive inhibition. Substance that resembles normal substrate competes with substrate for active site. Second is feedback inhibition. Concentration of product at the end of a pathway blocks the action of a K enzyme. Third is feedback repression. Inhibits at the genetic level by controlling synthesis of K enzyme. Fourth is enzyme induction. Enzymes are made only when suitable substrates are present. So we have heard a image of competitive inhibition. Energy, it is the capacity to do work or cause change. Endergonic reaction consume water, while the exergonic reaction release energy. Redox reaction always occur in pairs. There is an electron donor and electron acceptor which constitute a redox pair. The process salvages electrons under energy. Released energy can be captured to phosphorylate or ADP or another compound. Electron carriers resemble shuttles that are loaded and unloaded with electrons and hydrogens. Most carriers are coenzymes, the NAD, the FAD, and NADP, and the coenzymes A and a compound of the respiratory chain. The NAD reaction. We have heard the pictures. And electron carriers. So we have heard the ATP. Three-part molecule consisting of adenine, ribose, and 3-phosphate group. Adenine, a nitrogenous base, and the ribose is a 5-carbon sugar. And also we have the removal of the terminal phosphate releases energy. The ATP, the phosphorylation of glucose by ATP. The formation of ATP. First, the substrate level phosphorylation. Second is oxidative phosphorylation and the photophosphorylation. Substrate level phosphorylation. So we have here the substrate, the product, from substrate to product, and then down to ADP to ATP. Catabolism of glucose. The glycolysis, the tricarboxylic acid cycle, the Krebs cycle, respiratory chain, and electron transport. So we have heard the metabolic strategies. So glycolysis. We We have heard the overview of aerobic respiration, the glycolysis. Glycolysis is a glucose 6C is oxidized and split into two molecules of pyruvic acid or 3C. TCA process pyruvic acid and generate 3 carbon dioxide molecules. Electron transport chain accept electrons NADH and FADH, generates energy through sequential redox reaction called oxidative phosphorylation. So we have the glycolysis. This is cycle, the electrons transport system, chemiosmosis. Of course, we have the fermentation. Incomplete fermentation, it is incomplete oxidation of glucose or other carbohydrates in the absence of oxygen. Uses organic compounds as terminal electron acceptors. Yield a small amount of ATP, the production of ethyl alcohol by yeast acting on glucose, formation of acid, gas, and other products by the actions of various bacteria on pyruvic acid. So we have the fermentation. The product of fermentation, the pyruvate. From pyruvate, then we have the branches, the formic acid, the lactic acid, acetic acid, acetyl-methylic phenol, acid, the ethanol. Many pathways of metabolism are bidirectional or anti-bolic. Metabolites can serve as building blocks or the sources of energy. So that's it. So those lectures or the topic is all about the microbial